Hello, and welcome to MBio 3470 Exam Prep. Today we're going to do a quick review of gram-negative quorum sensing in Vibrio fischeri to help everyone prepare for the final. You'll also get to answer some questions along the way. Let's get started! As you all know, Vibrio fischeri has a symbiotic relationship with the Hawaiian Bobtail Squid, in which Vibrio fischeri uses quorum sensing to regulate bioluminescence in the squid. And the squid provides a host for Vibrio fischeri to colonize. In fact, quorum sensing was first discovered in Vibrio fischeri. Remember, the quorum sensing phenotype in this case is the bioluminescence, similar to how gram-positive bacteria like Staphylococcus aureus use quorum sensing for virulence. Now, when the population density of Vibrio fischeri is low, as it is when they're free living, the bacteria remain dark. But when the population density is high, as it is in the light organs in the Hawaiian Bobtail Squid, Vibrio fischeri emits a bluish-green light. Now you might be wondering, how does the population of Vibrio fischeri get so dense in the squid? Aren't there other bacteria competing with it? Well, there sure are. There are other species of gram-negative bacteria that aggregate in the mucus of the squid epithelial cells. However, Vibrio fischeri is able to outcompete all the others to become the one dominant species. There are two factors that make this possible. Can you name them? The first one is motility. Non-motile bacteria cannot colonize the light organ due to the microcurrents created by the cilia in the duct leading to the light organ. The second one is light production. Bacteria that can't produce light will not persist in the light organ. Now, let's get to the interesting part. We've talked about how Vibrio fischeri colonizes the squid. Now, let's talk about how it produces light. Do you know the name of the enzyme that's required for light production? Luciferase, of course. That was an easy one. Luciferase is an enzyme regulated by two genes, LuxR for regulator and LuxI for inducer. LuxR is a transcriptional activator that responds to autoinducer concentration in the environment. LuxI is a protein for autoinducer synthesis. Remember, autoinducers are small self-generating signaling molecules. Do you know the name of the autoinducer in gram-negative quorum sensing bacteria? That's right, N-acyl homoserine lactones, or AHLs. Another easy one. So, LuxI encodes AHL synthase, which is an enzyme that makes AHL. LuxI is always transcribed at a basal level. It is also positively regulated, which means that once it is activated, AHL synthesis increases rapidly. Now there are two domains of LuxR, one on the N-terminus and one on the C-terminus. Do you know which terminus binds to the DNA and which terminus binds to the autoinducer? Correct. The N-terminus is the autoinducer binding domain, while the C-terminus is the DNA binding domain. The C-terminus of LuxR binds to the Lux regulatory region and the RNA polymerase, and it activates transcription of the luminescence operon. However, if there isn't enough autoinducer present for the N-terminus to bind to, the N-terminus will interfere with the C-terminus. So the C-terminus can't bind to the DNA. Now, once LuxR has bond to AHL, what is the name of the regulatory region that it binds to next? The Lux box. The Lux box is a 20 base pair inverted repeat, located -42 base pairs from LuxI. The Lux box is required for autoinduction and bioluminescence. Once the LuxR bound to the AHL binds to the Lux box, transcription occurs, and luciferase is made. We talked about LuxR and LuxI, but what are the names and the order of the structural genes for luciferase biosynthesis? That's right, LuxCDABE. If you want help with that, just remember, can dogs actually boogie everywhere? And remember, there's another gene too, LuxG. whose function is unknown. Remember, Lugg's eye is always on at a basal level, but as the population of Vibrio fischeri becomes more dense, more AHL synthase is produced, which makes more AHL, which binds to Lugg's R, which binds to Lugg's box, which allows transcription of the operon to occur and luciferase to be made. Way to go! You're now an expert on quorum sensing in the Vibrio fischeri. Good luck studying, everyone!